Hey, what's going on, guys? Welcome back to The Last Viking TV. Now, what we're looking at here is going to be the coronavirus. And the coronavirus as opposed to the hantavirus. What are the connections in between these two? Well, the coronavirus belongs to a family known as Coronavirus. Okay, keep that in mind. Coronavirus. The word viridae, V-I-R-I-D-A-E, viridae. Okay, let's separate those two words for a second. And under a electron microscope, they look like spiked rings. They're named for these spikes, which form a halo crown. So the spike goes up and then creates a halo round on the top. And so that spike is the one that actually goes across and sticks itself to your insides. And then finally it sticks in and then it manifests. So it grows, right? And then it spreads. The thing about this virus is that it spreads so rapidly. Your body is able to recreate this virus multiple times so fast that your body cannot, your immune system, your T cells, are not able to produce enough T cells. Instead of producing more T cells, it produces more coronavirus, COVID, viridae cells. So, they're named for these spikes, which form a halo crown. Corona is Latin for crown. Around their viral envelope. Okay, so coronaviruses contain a single strand, okay, of RNA. You might want to remember that. Coronaviruses contain a single strand of RNA, as opposed to DNA, which is a double strand. So the helix, basically for DNA, goes crisscross like this and around each other. That's what creates our DNA. RNA is just a one strand. Okay? One strand. Not two. Okay, so coronavirus contains a single strand of RNA as opposed to DNA, which is double-stranded, within the envelope, and as such, can't reproduce without getting inside living cells and hijacking their machinery. The spikes on the viral envelope, the actual cell itself, has these spikes. So you got a cell here on the inside, then you got spikes on the outside. It cannot go through the body without these spikes, okay? In nature, natural occurring viruses, they're all round. They don't have spikes like this. Things that have spikes on them are designed, very carefully designed, and you can change their DNA on how they're produced. Like what we're doing with cloning animals and stuff like that. They can clone animals that have specifically brown eyes or blue eyes and may have longer horns on a cow or a bull than other cows. They can be very specific in genetics. And those are more complicated because they're double strand. How much easier would it be to work with a single cell structure that only has one strand? A lot easier. So they've specifically designed the algorithm of this virus. They designed the outside to specifically be able to attach itself to get this virus to spread like wildfire. In my previous video that is currently uploading right now, it's called Breaking News HANTAVIRUS. It's uploading right now. In that video, I talk about how there is, back in Vietnam War, there was over 3,000 people that contracted this hantavirus. not coronavirus, hantavirus. And what a coincidence that all 3,000 people would catch this virus. If this virus only comes, it's very rare that it would transfer from animal to animal to animal. Actually, very rare from animal to human. And then it would be even more rare and highly unlikely that it would transfer from human to human. Unless it was altered. Unless the structure of the cell itself was altered to specifically attack human structure cells. Do you follow that? I'll kind of go over it again. In Vietnam, there was over 3,000 soldiers, U.S. soldiers, that got infected with the hantavirus. Now, scientists and Democrats, they say that this virus only comes from animals to animals. It does not come from or affect animals to humans. Maybe on a rare cases, it may get one person infected if they're all up in the rat's bottom and just sniffing that stuff, right? You might get it that way. But explain me this, how did 3,000 soldiers get the coronavirus all on the same time? 3,000 of them were infected with the hantavirus. But then the scientists and Democrats also say that it's not transferable from human to human. I wonder how the humans got it. If it's not transferable from animal to human, first of all, that's a rare case, and then it's definitely not likely that it would ever transfer from human to human. But then explain, how did 3,000 people get this virus? How? Because the cell structure of this virus was altered. It was altered because it's easy and super cost effective to wipe out a whole entire army with a single gram of a biological weapon, an unseen weapon. These other countries like China, other places that we've fought against, they have an understanding for this biological weapons that the United States has no idea about. Or maybe the U.S. does have an idea about it, and they're just not telling us about this specific viruses. So, I've been doing a lot of research on this. As you can see, my portfolio on my channel here, you can go through almost 350 videos. 100 plus of them are going to be about the coronavirus. I've been in contact with some FBI officials, and those videos and informations are going to be coming out pretty soon. I've been in contact with the CDC. Those videos and recordings will be coming out pretty soon once I get a clearance from them. Of course, I can't release phone calls and stuff like that. I have to notify them, and I did notify them that it was going to be recorded. So I have to get clearance from them that it's okay for me to put out our conversation. So, of course, then that comes into a whole thing. So I'm waiting for that to be cleared before I put that on my YouTube channel. So now, moving on. The spikes on the viral envelope help the coronavirus bind to cells, which gives them a way in, like blasting a door open with C4. Once inside, they turn the cell into a virus factory. So again, this coronavirus is only as deadly as it is because of those spikes. Okay? In nature, out there in the wild, these cells, they don't exist. They don't. You look back at the cell structures of any other virus, it's rounded. You might have a little bit of fluff or furs on the outside, but they're rounded. None of them have spikes. Spikes. All right? Once inside, they turn the cell into a virus factory. The RNA and some enzymes use the cell's molecular machinery to produce more viruses, which are then shipped out of the cell to infect other cells. Thus, the cycle starts anew. Now, you've heard me talking about the hantavirus. The hantavirus, keep in mind, we're talking about single-cell RNA structures. This is the same exact one that we're going to be talking about that infected 3,000 plus military soldiers in Vietnam during the Vietnam War. Hantavirus. Hantaviruses belong to the, I'm going to call it the Beunia virus. Beunia virus. That's what it is. Beunia virus. Family of viruses. There are 5 generations within the family. Beunia virus, Phlebovirus, Norovirus, Tospovirus, and then Hantavirus. Okay, again, you can take a look at those viruses there. So each is made up of a negative sense single-strand RNA virus. I'll read that again. Each is made up of a negative-sensed single-strand RNA virus. Single-strand RNA. What did the coronavirus say? Coronavirus contains a single strand of RNA. We go back to hantavirus. Each is made up of a negative-sense single-strand RNA virus. Any connection there? We've been told that no other virus is like this except now. Now all of a sudden, here comes the hantavirus that magically infected over 3,000 U.S. soldiers back in Vietnam War. And it's classified as a biological weapon that is still till this day not known or recognized. Specifications and classified information is obviously not available to the public. So then again, you have to use your best judgment on whether you think that this is possible. In my opinion, I strongly think and feel and believe 100% without a doubt, because nothing has come across my research to show or contradict any of this information, that it's not a biological weapon and that we were not under attack from China, a communist country that is seeking global dominance, stemming from the trade war mid last year. Like other members, okay, so hold on. All these genera, including arthropod-borne viruses, with the exception of the hantavirus, which is it says rodent-borne, but again, this hantavirus, this form of hantavirus, obviously altered. You have to take in some of the facts that they say, base them on where the history of these viruses. So yes, it may have came from a rodent. Okay. They say that the hantavirus is only from rodents to rodents, that it cannot go from rodents to humans. First lie. The second lie is that it's impossible. Again, it is impossible for the hantavirus to go from rodents or animals to humans. That's the second lie. Third lie. They say it is impossible for it to go from human to human. 3,000 soldiers getting infected, all on the same mission within the same time period in a Vietnam War. Third lie. Fourth lie, indirectly. They say nothing is like this coronavirus because it has spikes. coronavirus is the same structure as the hantavirus. Same structure. In my previous video that's uploading here, called Breaking News, Hantavirus, New Virus, you will see that it has spikes on it just like the coronavirus. So fourth lie. How many more lies do you need to see the truth? So like other members of the Beunovirus family, hantaviruses are enveloped with viruses with a genome that consists of 3 single-stranded RNA segments, designed S small, M medium, and L large. All hantaviral genes are encoded in the negative genome complementary sense. So, I mean, does it already kind of like it's so there's 3 different versions of this hantavirus. There is a small structure, a medium structure, and a large structure, and just depending on which 3 of those structures you get infected with depends on really the severity of the virus. If you get infected with a large cell structure, most likely you're probably going to have harder times, but then the production rate. So I mean, a lot of those things go into I'm not going to get into all that stuff right now, but there is a light cold, medium symptoms of a cold, and severe ICU symptoms of a cold. But it's all a cold. So the same thing with this. They're all made up of single cell structures, small, medium, and large. All hantaviral genes are encoded in a negative genome complementary sense. The sRNA encodes the nucleocapsid in protein. The mRNA encodes a polyprotein that is contraslationally cleaved to yield the envelope glycoproteins G1 and G2. When you look at the cell structures of the coronavirus COVID-19, you will also see that the glycoproteins are also mentioned in the cell structure of the coronavirus. The RNA encodes the L protein, which functions as So now going back, the mRNA encodes a polyprotein that is contrastationally cleaved to yield the envelope glycoproteins G1 and G2. The lRNA encodes the L protein, which functions as a viral transcriptase replicase within virions. The genomic RNAs of the hantavirus are thought to complex with the N protein to form helical nucleocapsids, which circularize due to sequence complementary between the 5 and 3 terminal sequences of each genomic segment. Wow. If any of that information, I mean, if you're a biologist, okay, and you want to do an interview about this information, please leave a comment down below and I will do my best to contact you. Give me a Twitter, whatever, Facebook. I will find a way to contact you and we will do a face-to-face interview if you agree that this has a connection with the coronavirus and that this is possibly the structure and the design of something far more intelligent than nature itself. I'm talking about human intervention. Okay? Human altering of this cell to make it even more deadly than it originally is supposed to be. Because again, it's not supposed to come from animals. It's not supposed to infect from animals to humans. And it's not supposed to infect from human to human. Lie after lie after lie. I really want to get down to the truth of this to be able to help my viewers understand exactly what this is all about. The hantavirus replicate exclusively in the host cell cytoplasm. Entry into the host cells is thought to occur by attachment of virions to cellular receptors and subsequent endocytosis. endocytosis. Nucleocapsids are introduced into the cytoplasm by pH-dependent fusion of the virion with the endosmal membrane. Transcriptions of the viral genes is initiated by association of the L protein with the 3 nucleocapsids species. In addition to transcriptase, the replicase functions of the viral L protein is also thought to have an endonuclease activity that cleaves cellular messenger RNAs, mRNAs, for the production of capped primers used to initiate transcription of the viral mRNAs. As a result of this cap snatching, the mRNAs of the hantavirus are believed to be capped and contain non-templated 5 terminal extensions. The viral N and L mRNAs are thought to undergo translation at free ribosomes, whereas the mRNA is translated in the endoplasmic reticulum. G1 and G2 glycoproteins form heterodimers and are then transported from the endoplasmic reticulum to the Golgi complex, where glycosylation is completed. The L protein produces nascent genomes by replication via a positive sense RNA intermediate. Hantaviruses, hantavirans, are believed to form by association of nucleocapsids with glycoproteins embedded in the membranes of the Golgi, followed by budding into the Golgi cisternae. Nascent virions are then transported in secretory vesicles to the plasma membrane and released by exocytosis. So, with that being said, we've definitely connected. I mean, it doesn't take a genius to look at the images that we had before. If we just go ahead real fast here, I'm just going to go ahead and look at the coronavirus cell structure. Okay? So now we're going to take a look at, I think this is the best image here. So here we're going to see the spike glycoprotein, which are those purple little dots there. That is the glycoprotein. This is the coronavirus. Okay? The hemagglutinin acetylsterase glycoprotein, also the second little yellow-orange dot below the purple dot, and then the membrane glycoprotein, small envelope glycoprotein, the nucleocapsids, phosphoprotein. So then you have these structures on the inside. You see how they're wrapped. Okay, so they're wrapped and they're like little lines, little wires that are inside. This specifically, just looking at it, this is not something that nature would create. Okay, these little wires with little connections and little, it looks like a computer board. It really looks like a computer board, does it not? It looks like a computer board. It does not look like anything from nature. Next, we're going to go ahead and we're going to look at, let me see if I can actually, can I zoom in? No, I can't zoom in. That's about as zoomed in as I can get. Oh, here we go. Okay. So non-infected cell. There we go. So there's a coronavirus cell. That is the natural coronavirus cell. Now the normal cell, non-infected cell of a coronavirus is going to look like the one that we have over there. The coronavirus infected cell, obviously the altered cell by human alteration, looks like the one right above here, where it has the LC3 stage 2 and LC3 stage 1. You're going to see that there's sequences within there. Okay? Now, going on, we're going to take a look at the hantavirus. Hantavirus cell structure. Now, looking at the hantavirus cell structure, we're going to take a look at something that is a little bit more. So I really want to see the glycoprotein. Here we go. So here we're going to see the RNA polymerase has the same glycoproteins on the outside, the same spikes that are in the coronavirus cell. So again, here we go. We're going to go right back to here. So here we have the spiked glycoproteins and the hemagglutinin glycoprotein. We're going to go here. We're going to see glycoprotein, RNA polymerase, and glycoprotein 2. We see stage 1 and stage 2 of the glycoprotein. Here we see the stage 1 and stage 2 of the glycoprotein. So there's 3 different cell structures of this on the inside, which are small, medium, and large. And then if we keep going on, we'll be able to see the cell structure of the actual endocytosis, stage 2 up there at the top, and then egrase right here closest to my edge of my finger, egrase number 7. And then you're going to see the assembly at plasma membrane going from the inside to the outside. Looks like a little zit, like a little bump. So right below that is the membrane. Again, this is more complicated than the actual. I mean, so basically they took the hantavirus. They took the hantavirus. They made it more complicated, added bigger spikes to it, and made it highly more human-friendly virus. Okay? Because it just wasn't affecting enough people at that time. So what we're dealing with here now is the hantavirus. It has a 50% chance mortality rate, which is the reason why we're seeing an increase spiked in death rates. Okay? If this is all making sense to you, please drop a subscribe down below and share. So as we move on to this hantavirus, we're going to see more and more commonality with the structures of the coronavirus. Now, you see this wire on the inside? This wiry structure on the inside? You've got a large, a medium, and a small. Okay? The hantavirus, we're going to go through a couple other clips. Schematics of the hantavirus morphology. So we got the glycoprotein GN, G1, glycoprotein GC, G2, RNA polymerase, RD, RP, and then nucleocapsid proteins forming helical nucleocapsids with the viral ssRNA segments S, M, and L. So you can see the structures are that of the coronavirus that we're working with right now. So again, this is kind of a 3D print of that. We're looking at, so before they were saying, that we can't get this virus, but now they are saying that we can, and they're very similar to what you would feel with the coronavirus. Chills, fevers, muscle aches, dry cough, headaches, nausea, vomiting, shortness of breath, acute respiratory distress, serious infections that quickly worsen. And it fills up your lungs. So again, here we go. You see, here's the spikes. Spikes for antivirus. Hantavirus, yeah, hantavirus. So here we go. Protein coat, nucleic acid on the inside, spikes for attracting to specific cell structures, and an envelope. And then you look at the coronavirus, it just seems like a new, updated version of the hantavirus. This specific type of hantavirus has spikes. Okay? It's right there. Right there. Hantavirus. Coronavirus. 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 We look at the hantavirus. Hantavirus. Okay? You can see these little, you can see right here, if you can see it right here, right up here, everything that I'm looking at, my search, is all hantavirus. Okay? And then let's see if I have it over here. Hantavirus, all up here on the top. Hantavirus, hantavirus. You can see it. It's hantavirus. So the hantavirus that we have here, same exact cell structure as the coronavirus, with the E protein, S protein, and M protein. 3-stage protein and 3-stage protein. What more information do you want? Please subscribe, please share, and thanks for watching. Until next time, The Last Viking TV is out. I got more content coming. Thanks for watching.